This video summarizes our paper describing the Type 2 Diabetes Knowledge Portal that was published on April 4th. Associations between human genetic variation and clinical phenotypes have become a foundation of biomedical research. Most repositories of these data seek to be a disease-agnostic and therefore lack disease-focused views. The resource was developed to address two gaps in our capabilities. The first gap is that many disease-specific datasets are too narrow in scope. The second gap is many analyses can only be conducted after data from disparate resources are combined. This resource addresses these gaps by aggregating and integrating data and methods of many types while focusing on only one disease area. It contains genetic association summary statistics, genomic annotations, bioinformatic method results, and expert knowledge for T2D and related traits. Our web interface includes a set of core pages for browsing these results in the vicinity of a gene, variant, or a region of interest. We also include a suite of complex tools for expert users. As of October, the site contained 382 GWAS full summary statistic datasets. Comparing across three other disease-agnostic resources, our resource has the greatest representation of full summary stats. Since 2015, we have the greatest representation of datasets with over 10,000 samples, including 71 unavailable elsewhere. 31 summary statistic datasets not included in the site were either small, old, or largely overlapping with other studies. Our datasets skew heavily toward European samples due to the ethnic and socio-demographic biases of GWAS, although ethnic diversity has increased somewhat over time. We employ novel methods for a meta-analysis, including a bottom-line approach. We compared the loci with fasting glucose associations in the resource to those in the GWAS catalog in the Open GWAS project. As a result, we both add and omit glycemic trait associations relative to the GWAS catalog. As of October, we have 5,418 genomic annotation datasets. These are stored in the Common Metabolic Diseases Genome Atlas, a sister resource connected to our site through a series of REST APIs. For non-geneticist users, we outline the following workflow to help researchers evaluate and interpret genetic results. If users would like a list of genes with the strongest genetic support for T2D or related traits, then they should use the Predicted Effector Genes tool. This tool contains 4 lists of genes predicted to mediate GWAS associations in the T2DKP, categorizes genes based on the strength of these predictions, and allows users to see evidence underlying each prediction. We offer several different interactive tools for those who want to explore the data in detail. In addition, the paper offers examples of using the tools and best practices for interpreting the results. Our resource aims to make such supporting evidence readily available and interpretable in a T2D-specific resource, thereby promoting the understanding and treatment of diabetes and its complications. We provide one potential model for sharing information derived from sensitive datasets in a manner that is broadly accessible but is still useful to disease experts. Our resource also suggests a means to encourage researchers to share data and results in settings where they have been traditionally hesitant. If you are interested in using the T2DKP, please scan the QR code or visit type2diabetesgenetics.org.